this is Smile and my partner Minkai.
Hello everyone.
Today we will be discussing one developed glycoproteomics technique for profiling N and O glycopeptides from whole cell proteomes called IsoTAG.
Okay, and I will cover the introduction and the method part, and my partner will cover the result and discussion part. So now let's start.
Okay, now let's start. This article was published from the April 2017, which is a very new one. And although the score is only 8, but it's a very latest article, so I think it's pretty good. For the IsoTAG, it's termed as isotope-targeted glycoproteomics. And for the author information, the most of them are come from Stanford University, and the major author, Christina. She had published several similar articles before about this technique, IsoTAG. And first of all, I just want to introduce glycosylation. So glycosylation is a post-translation modification that generates extreme diversity and contributes to a plethora of cellular functions such as modulating protein structure, stability, and recognition. Well, the dysregulation of glycoprotein is often correlated with disease states, including cancer and inflammation. And now there are 3 main types of glycoprotein. The most common types are the nitrogen-linked and oxygen-linked. Well, the carbon-linked is very rare. For the nitrogen-linked, it will the nitrogen atom on the side chain of aspirin. For the oxygen linkage, it will the oxygen atom on the side chain of styrene. So the ability to define the intact glycoprotein would accelerate structure-function studies and serve to identify potential disease biomarkers and therapeutic targets. Well, there has been much interest in profiling the intact glycoprotein. The complexity of glycoproteoforms remains challenging to completely define, and now the studies are limited to nitrogen glycans. For the oxygen glycan, there are no proper sequence and specific enzymes. So, how to profile? The mass spectrometry is commonly used to do the profile, and there are 2 requirements. First is enrichment of specific glycoprotein subtypes, and which can be done by metabolic labeling. And the second one is reduction in glycan heterogeneity. And for the nitrogen glycoproteins, it can be done by enzymatic digestion with PNGSF or EndoH. And for the oxygen glycoproteins, we use genetic engineering in cell lines. So our design study is use the platform isotope-targeted glycoproteomics to do the isotopic recording and enrichment, and then do the tandem MS and intact glycopeptide assignments. And here is a method. 15 cell lines were derived from various human tissues, and they were treated with sugar labels to generate metabolically labeled glycoproteins. The glycoproteins were tagged with IsoTAG ProbMixture 3, and we found in this article the ProbMixture 3 is the best one, and now it happens chemical conjugation. And now after the trypsin digestion and the cleavage of ProbMixture 3, we covered isotopically recorded glycopeptides for analysis. And then the glycopeptides were analyzed by targeted LC-MS using IsoStem version 2 to direct selection of isotopically recorded species. And a survey of 15 human cell lines was performed to demonstrate the broad applicability of IsoTAG and reviewed 74 glycan structures over 3,500 intact glycopeptides. And here is the workflow for model generation and pattern matching by IsoStem version 2. And for the A is the full scan MS data, abandoned across scans, and picks are selected for analysis by the charge state. We just choose top 3 picks. And for the B is a modern generation. And a predict distribution is generated by convolution of a base pattern derived from a modified average model and an overlaid tag pattern. And compare the A and B, and we can get C. Observed and predict distributions are compared by subtraction to obtain the linear error and the MSQR errors. And here is the modality of pattern recognition by the IsoTAG, and here is the model system. We are using BSA, and this was tagged with halogen tags, digest, and spec into Jurkit lysite. And there are 3 different tags. Tag 1 is monobromide, tag 2 is dichloride, tag 3 is dibromide. And from the C, and we can find the FDR rate. For the version 2's FDR rate is lower than the version 1, and which can be found that IsoTAG version 2 is better than the version 1. And from the D is the score distribution of pattern matches produced by IsoTAG version 2. Searching for halogen tagged peptides in complex mixtures. Jurkit lysate with halogen tagged BSA peptides generate better score pattern match than the Jurkit lysate alone. So the halogen tagged peptides are better scored. The score is higher, this means it's better match. And now I will just let my partner Mile to do the result and discussion part.
Although the dibromide tag was highly recognizable with IsoStamp 2, alternative probes with smaller and less sterically congested structures were explored. The primary motivation was the absence of core fucosylation in N-glycans from their initial studies. This absence was potentially caused by lack of access of the probe to the sterically congested GLACNAs or the N-acetylglucosamine tetracylated. The probeness hypothesis, a series of IsoTAG probes encoded by 0, which is the N, here, 2, which is the M plus 2, and 4, which is the M plus 4 deuterium atoms. Probe mixtures are mixtures of non-deuterated and deuterated probes. For probes 1 to 4, it is a mixture of 0 and 2 deuterium atoms, while for the 5 to 8, it is a mixture of 0 and 4 deuterium atoms. And based on the results that we got, probe mixture 3, which is this one, gave the highest true positive result. The use of probe mixture 3 in studying the 15 cell lines resulted in the identification of 55 N-glycans, which is here, which were grouped into 5: the high mannose, the hybrid, the biantenary, the triantenary, and the tetraantenary. And they also identified the total of 18 all-glycans. The high mannose ones are the most abundant for the N-glycans, while the all-GLACNAC or the acetylglucosamine are the most abundant ones for the all-glycans. The protein localization, labeling, and frequency of the identified glycoproteins and glycopeptides were also studied. In this graph here, we could see that approximately more than 60% of the proteins are intracellularly localized. The galactosamine and myosin labeling produced more all-glycans, which is here. And the frequency is mostly concentrated in one cell line. Table 1 summarizes the top 10 most frequently observed glycopeptides. and the most abundant ones are the ones with the high malose. IsoTAG was compared with per-methylated N-glycan analysis using DMSO or the dimethyl sulfoxide. PC3 line or cell line was used and analyzed by Maldi-Toftoff. It can be seen here that most of the glycans observed using DMSO were also detected by IsoTAG, which are marked with the asterisk here. The block here indicates the intact glycans detected in both analyses. Based on the results they have obtained, they have observed that galactosamine labeling incorporates only one azido sugar per glycopeptide, while the mannose label uses up to 4 azido sugar substitutions. Aulactans is the primary glycan present with more than 60% abundance. Carfucosylation and galactose incorporation were observed at the reducing end of the glycan structures, supporting the hypothesis that Prov mixture 3 would be able to tag a broader spectrum of N-glycan structures. Lastly, native glycans carried homologous glycan structures to those identified by IsoTAG, irrespective of metabolic labeling. In conclusion, the platform offers a high-fidelity pattern matching and characterization of intact glycopeptides by the bromine tag peptides evaluation. Isotopic recoding uniformly improved the selection and confidence of results in all samples tested. And the platform accelerates the structure function studies of the glycogen. For future applications, the methods can be optimized to analyze the azide-incorporated glycan structures directly. It can also be applied to discern glycosylation in a site-specific manner and further correlation with structure to function for particular glycosides. It may also be used to study other chemically tagged post-translational modifications. And that's all. Thank you.